Hi, and welcome to part 2 of the EDGE tutorial series. In part 2, we dedicate one video to each EDGE module. In this video, I will demonstrate how to access EDGE through a web browser. After this tutorial, you will know how to set up a new user account, log in, and set up an EDGE run. After going into user management, we will set up two EDGE runs together. One using FASTQ files, and the other using public data pulled from the SRA database. When running EDGE, the following modules are turned on by default: pre-processing, assembly and annotation, and taxonomy classification. The following videos will shed light on what processes and analyses are useful for a given dataset. Let's go ahead and switch to the live view of EDGE. Open a web browser and go to the following address. This is the outward-facing version of EDGE hosted by the Naval Medical Research Center. On the home page, you can see information covered in part 1 of this series. Feel free to look around at the overview, EDGE features, basic implementation information, source code, and read the docs tutorial links. If you look at the top right, we can click on a silhouette and create a new user account. This is where you will log in the future. For an account, new users will need to contact the Naval Medical Research Center's development team. You can use this link here. You will receive an email when your account is created. Once it's created, you can click on the silhouette and log in. To initiate the EDGE workflow, scroll to the top left and click on Run EDGE. This interface contains all of the EDGE modules to analyze your sample. Let's go ahead and set up a project called EDGE Tutorial FASTQ. Note there is a 30-character limit in the project name. Avoid using spaces, but dashes and underscores are fine. The description is optional, but useful if you start submitting a lot of jobs. You can see here that EDGE accepts single and paired-end data. Our example is paired. Click on the circular button on the right of Pair 1 FASTQ file. Select the data under PublicData, testData, scroll to the bottom and find the E. coli 10x.1 FASTQ. Let's do the same for Pair 2. This is going to be the .2.FASTQ. If an EDGE server were set up locally in your institute, your data can be linked to these drop-down lists for easy access and analysis. This data is now ready to be analyzed. Let's open up another tab with the same demo address and set up a run using data deposited into SRA. We're at the same hobonickel.getedge.org under Run EDGE. We're going to take a look at a patient fecal sample from the 2011 E. coli outbreak. If you check the NCBI SRA website, you will see that this particular sample is a metagenomic sample with paired-end reads from an Illumina HiSeq machine. Let's go ahead and name this project EDGE Tutorial SRA. We're also going to give it a description. To get data from SRA, click Yes on the input from NCBI SRA. Then add the run information. I already gave you that number here. Just for fun, let's go ahead and click Submit at the bottom of each of these pages. The goal of this is to demonstrate that EDGE default parameters are relatively robust and you can successfully run EDGE without changing anything. Scroll to the bottom and click Submit. We'll go back to the E. coli FASTQ sample, scroll to the bottom, and also just click Submit with all the default parameters remaining the same. Alright, that's it. In the next video, we're going to go into detail about pre-processing.